Replicating and repairing DNA. Reproduction is a fundamental property of all living systems. The process of reproduction can be observed at several levels. Organisms duplicate by asexual and sexual reproduction, cells duplicate by cellular division, and the genetic material duplicates by DNA replication. The machinery that replicates DNA is also called into action in another capacity to repair the genetic material after it has sustained damage. These two processes, DNA replication and DNA repair, are the subjects addressed in this chapter, which we will be learning with the help of multiple video sections. Video sessions. The capacity for self-duplication is presumed to have been one of the first critical properties to have appeared in the evolution of the earliest primitive life forms. Without the ability to propagate, any primitive assemblage of biological molecule would be destined for obliteration. The early carriers of genetic information were probably RNA molecules that were able to self-replicate. As evolution progressed and RNA molecules were replaced by DNA molecules as the genetic material, the process of replication became more complex, requiring a large number of auxiliary components. Thus, although the DNA molecule contains the information for its own duplication, it lacks the ability to perform the activity itself. As Richard Lewontin expressed it, the common image of RNA as a self-replicating molecule is about as true as describing a letter as a self-replicating document. The letter needs a photocopier. The DNA needs a cell. Let's see then how the cell carries out this activity. First process, DNA replication. The proposal for the structure of DNA by Watson and Crick in 1953 was accompanied by a suggested mechanism for its self-duplication. The two strands of the double helix are held together by hydrogen bonds between the bases. Individually, these hydrogen bonds are weak and readily broken. Watson and Crick learned that replication occurred by gradual separation of the strands of the double helix, much like the separation of two halves of a zipper. Because the two strands are complementary to each other, each strand contains the information required for construction of the other strand. Thus, once the strands are separated, each can act as a template to direct the synthesis of the complementary strand and restore the double-stranded state. Then, semi-conservative replication. This made some predictions. That is, Watson and Crick proposed model made some predictions concerning the behavior of DNA during replication. According to the proposal, each of the daughter duplexes should consist of one complete strand inherited from the parent duplex and one complete strand that has been newly synthesized. Replication of this type is said to be semi-conservative because each daughter duplex contains one strand from the parent structure. In the absence of information on the mechanism responsible for replication, two other types of replication had to be considered. In conservative replication, the two original strands would remain together after serving as templates, as would the two newly synthesized strands. As a result, one of the daughter duplexes would contain only one parental DNA, while the other daughter duplex would contain only one newly synthesized DNA. In dispersive replication scheme, the parental strand would be broken into fragments and the new strands would be synthesized in short segments. Then the old fragments and new fragments would be joined together to form a complete strand. As a result, the daughter duplex would contain strands that were composites of old and new DNA. At first glance, dispersive replication might seem like an unlikely solution, but it appeared to Marx Delbook at the time as the only way to avoid the seemingly impossible task of unwinding two intertwined strands of DNA duplex as it replicated. To decide among these three possibilities, it was necessary to distinguish newly synthesized DNA strands from the original DNA strands that serve as templates. This was accomplished in studies on bacteria in 1957 by Matthew Michelson and Franklin Stahl in the California Institute of Technology, who used heavy 15N and light 14N isotopes of nitrogen to distinguish between paternal and newly synthesized DNA strands. These researchers grew bacteria in medium containing 15N ammonium chloride as the sole nitrogen source. Consequently, the nitrogen containing bases of DNA of the cells contained only the heavy nitrogen isotopes. Culture of heavy bacteria were washed free of the old medium and incubated in fresh sodium with light. 14N containing compounds and samples were removed at increasing intervals over a period of several generations. DNA was extracted from the samples of bacteria and subjected to equilibrium density gradient centrifugation. In this procedure, the DNA is mixed with a concentrated solution of cesium chloride and centrifuged until the double-stranded DNA molecule reach equilibrium according to their density. In the Michelson-Stahl experiment, the density of a DNA molecule is directly proportional to the percentage of 15N or 14N molecule it contains. If replication is semi-conservative, one would expect that the density of DNA molecule would decrease during culture in the 14N containing medium in the manner shown in the upper set of centrifuge tubes. After one generation, all DNA molecules would be 15N 14N hybrids, and their buoyant density would be halfway between that expected for total heavy and total light DNA. As replication continued beyond the first generation, the newly synthesized strands would continue to contain only light isotopes, and two types of duplexes would appear in the gradients, those containing 15N and 14N hybrids, and those containing only 14N. As the time of growth in the light medium continued, a greater and greater percentage of the DNA molecule present would be light. However, as long as the replication continued semi-conservatively, the original heavy parental strand would remain intact and present in hybrid DNA molecules that occupied a smaller and smaller percentage of the total DNA. The results of the density gradient experiments obtained by Michelson and Stahl is very important, and they demonstrate unequivocally that replication occurs semi-conservatively. The results that would have been obtained if replication occurred by conservative or dispersive mechanism are indicated. By 1960, replication had been demonstrated to occur semi-conservatively in eukaryotes as well. The original experiments were carried out by J. Herbert Taylor of Columbia University. Cultured mammalian cells were allowed to undergo replication in bromodioxyuridine, a compound that is incorporated into DNA in place of thymidine. Following replication, a chromosome is made up of two chromatids. After one round of replication in BRDU, both chromatids and each chromosome contained BRDU. After two rounds of replication in BRDU, one chromatid of each chromosome was composed of two BRDU containing strands, where the other chromatid was a hybrid consisting of BRDU containing strand and a thymidine containing strand. The thymidine containing strand had been repairing a part of original parental DNA molecule prior to addition of BRDU to the culture. Then replication in bacterial cells. We will focus in this section of the chapter on replication in bacterial cells, which is better understood than the corresponding process in eukaryotes. The early process in bacterial research was driven by genetic and biochemical approaches, including the availability of mutants that cannot synthesize one or another protein required for the replication process. The isolation of mutants unable to replicate their chromosome may seem paradoxical. How can cells with a defect in this vital process be cultured? This paradox was slowed by the isolation of temperature-sensitive mutants, in which the deficiency only reveals itself at an elevated temperature, termed the non-permissive or restrictive temperature. When growth at the lower permissive temperature, the mutant protein can function sufficiently well to carry out its required activity, and the cells can continue to grow and divide. Temperature-sensitive mutants have been isolated that affect virtually every type of physiological activity, and they have been practically important in the study of DNA synthesis as it occurs in replication, DNA repair, and genetic recombination. The development of in vitro systems in which replication can be studied using purified cellular components. In some studies, The DNA molecule to be replicated is incubated with cellular extracts from its specific proteins suspected of being essential have been removed. In other studies, the DNA is incubated with a variety of purified proteins whose activity is to be tested. Taken together, these approaches have revealed the activity of more than 30 different proteins that are required to replicate the chromosomes of E. coli. In the following pages, we will discuss the activities of several of these proteins whose functions have been clearly defined. Replication in bacteria and eukaryotes occur by very similar mechanisms, and thus most of the information presented in this discussion of bacterial replication applies to eukaryotic cells as well. So replication forks and bidirectional replication. Replication begins at a specific site on the bacterial chromosome called the origin. The origin of replication on the E. coli chromosome is a specific sequence called ORIC, where a number of proteins bind to initiate the process of replication. Once initiated, the replication process proceeds outward from the origin in both directions, that is bidirectionally. Pair of replicated segments come together and join the known replicated DNA are termed replication forks. Each replication fork corresponds to a site where the parental double helix is undergoing strand separation and nucleotides are being incorporated into the newly synthesized complementary strands. The two replication forks move in opposite directions until they meet at a point across the circle from the origin where replication is terminated. The two newly replicated duplexes detach from one another and are ultimately directed to two different cells. Then unwinding the duplex and separating the strands. Separation of the strands of a circular helical DNA duplex poses major topological problems. To visualize the difficulties, we can briefly consider an analogy between a DNA duplex and a two-stranded helical rope. Consider what would happen if you placed a linear piece of this rope on the ground, took hold of the two strands at one end, and began to pull the strands apart, just like a DNA is pulled apart during replication. It is apparent that the separation of the strands of double helix is also a process of unwinding the structure. In the case of rope, which is free to rotate around its axis, separation of the strands at one would be accompanied by rotation of the entire fiber as restricted the development of tension. Now consider what would happen if the other end of the rope were attached to a hook on a wall. Under these circumstances, separation of two strands at the free end would regenerate increased torsional stress in the rope and cause the unseparated portion to become more tightly wound. Separation of the two strands of circular DNA molecule or a linear molecule that is not free to rotate. It is the case with large eukaryotic chromosome. It's analogous to having one end of a linear molecule attached to a wall, and all the other cases, tension that develops in the molecule cannot be relieved by rotation of the entire molecule. Unlike a rope, which can become tightly unwound, an overwound DNA molecule become positively supercoiled. Consequently, movement of the replication fork generates positive supercoils in the unpredicted portion of the DNA ahead of the fork. When one considers that a complete circular chromosome of E. coli contains approximately 4,100 turns and it is replicated by two forks within 40 minutes, the magnitude of the problem becomes apparent. It was noted earlier that cells containing enzymes called topoisomerases that can change the state of supercoiling in DNA molecule. An enzyme of this type is called DNA gyrase. A type 2 topoisomerase relies the mechanical strain and builds up during replication. In E. coli, DNA gyrase molecule travels along the DNA ahead of the replication fork, removing positive supercoils. DNA gyrase accomplishes this feat by cleaving both strands of DNA duplex, passing a segment of DNA through the double-stranded break to the other side, and then sealing the cuts, a process that is driven by the energy released during ATP hydrolysis. Next topic will be led with the help of next video.